The MDX utrophin double knockout mouse model is a very important mouse model for modeling Duchenne muscular dystrophy and other muscular dystrophies, due to the similarities with human DMD reports and the much higher severity reported in these mouse models. It has been an important and valuable model for clinical studies to combat and treat this disease. Here, I will present a brief history on the discovery and development of the MDX utrophin double knockout mouse line. The beginning dates back to the first discovery of the MDX model, created accidentally by Graham Bullfield in 1984. Bullfield was studying mutagenesis of enzyme activity in glycolysis pathways, meaning that he was trying to evolve newer and better proteins for glycolysis, and in particular was studying the enzyme pyruvate kinase, the enzyme involved in the final step of glycolysis, converting phosphoenol pyruvate to pyruvate and generating ATP. He was inbreeding mice to produce mutations and was screening for pyruvate kinase activity when he noticed a particular breed had threefold increased activity. Finding this unusual, he screened these mice for creatine kinase levels and performed histological examinations and found results consistent with reported results in human muscular dystrophies. Based on pedigree and linkage analysis, it was determined that the MDX mutation was X-linked, and crossing of MDX mice with 4 other X chromosome mutants revealed the location of the MDX mutation. So that was cool. There is now a mouse model with an X-linked mutation that phenotypically resulted in what was similar to human muscular dystrophies, albeit at a much milder severity. It had been known at the time that DMD was an X-linked recessive disease, but the best model that existed at the time was the DY mouse, which was an autosomal recessive, and it was not known whether the origins of the dystrophic phenotype were myogenic or neurogenic. So this new MDX model was already better. Later on, a man by the name of Hoffman came along and, in 1987, was able to isolate the dystrophin protein using polyclonal antibodies and showed that the MDX model, like in human DMD patients, lacked dystrophin. Soon after, another man came along, another researcher by the name of Piotr Szysinski in 1989, and he determined the explanation for the lack of dystrophin in MDX, which was a spontaneous nonsense point mutation resulting in a premature stop codon in exon 23. He did so by PCR amplification and sequence analysis. Now, with all these discoveries and tests done on the MDX model, it was strange that the MDX model itself was not very good to model for the phenotype of Duchenne muscular dystrophy. MDX mice have quite mild phenotypes, and it isn't until well into adulthood that mice show severe histological lesions as in human DMD muscle biopsies. So then came along 2 independent researchers, Anne DeConninck and Ronald Mark Grady, both of whom were, in 1997, working on the study of utrophin. It had been known that utrophin was found at the neuromuscular junction and co-localized with acetylcholine receptors, and it was thought to be particularly important in neuromuscular junction differentiation during development and muscle regeneration. Both DeConninck and Grady created these utrophin homozygous deficient mice by introducing a recombinant plasmid into embryonic stem cells and implanting the transfected cells into blastocysts. Using homologous recombination, they quite literally knocked out the native utrophin gene and replaced it with a dysfunctional version. In DeConnick's research, she disrupted the actin binding domain such that only the largest isoform was inactivated. In Grady's research, the cysteine-rich region of utrophin was disrupted, leading to no functional isoforms of utrophin. Furthermore, it had been known that utrophin had very similar structure and function to dystrophin, and was thought to perform redundant functions. So Tinsley showed, in 1996, that upregulation of utrophin in MDX mice alleviated histological lesions and reduced the dystrophic phenotype. As a natural result, both DeConninck and Grady, who had created their own unique lineages of utrophin-deficient mice, crossed their utrophin-deficient mice with MDX mice to produce 2 different types of MDX utrophin double knockout mice. These mice seemed like a better model for DMD due to their more dystrophic phenotype, which was more severe than in regular MDX mice. It turned out that the lack of utrophin, combined with the lack of dystrophin, resulted in better mouse models.